Now the question is, what controls in the direction of the transport? Whether it should move to the cytoplasm or it should move to the nucleus. There is a protein, and the name of that protein is Ran. Ran is a protein, and this Ran protein has the capacity to bind with the caryopherids. It can bind with the importance and it can bind with the exportins. So depending on that, it is able to move the cargo or direct the cargo either to the cytoplasmic side or to the in the nucleus itself. When we look closely at Ran, Ran is present in the cytoplasm as well as in the nucleus. Now Ran can bind to GTP as well as GDP. Ran bound to GDP is found in the cytoplasm, and Ran bound to GTP is found in the nucleus. Whether it is bound to GDP or GTP, that depends on enzymes that are present in these locations. In the cytoplasm, there is an enzyme called Ran-GAP. Ran-GAP stands for Ran-GTP. GTP is activating protein. GTP is activating protein. In the nucleus, there is another enzyme that is Ran-GEF. Full form of GEF is guanosine nucleotide exchange factor, that is GEF, which is able to replace a GDP with a GTP. What really happens is an interesting mechanism. Now let us begin with this molecule, that is if you have a Ran, so this is a Ran, and Ran is bound to a GDP molecule. So we will represent a GDP like this, and this is in the cytoplasm. Okay, it is in the cytoplasm. In the cytoplasm. Now, if you consider this is in the nucleopore, what happens is this will come inside. The Ran-GDP will come inside. Ran-GDP is here. If the Ran-GDP, as we have seen, if the GEF can act on it, if the GEF will act on it, and so this is the Ran-GEF will act on it, and it will replace this GDP for a GTP molecule. So GTP is added and the GDP is removed. So what effectively happened to Ran? Now he's a Ran that has got a triphosphate. So this is a Ran with a triphosphate attached. Now Ran with a triphosphate attached, it will come out. Okay, so little correction here. This is our nucleus. This is a nucleopore. Okay, nucleopore, and this will come out. Once this is out, this is now the Ran with the GTP attached. Ran with the GTP attached. Now once the Ran is with the GTP attached, this is converted to Ran-GDP. What is to be done? This GTP has to be converted to a GDP molecule. That is why GAP enzyme will convert Ran-GTP into a Ran-GDP molecule. Look at the mechanism quickly. So we began with the Ran-GDP molecule, and this is transported to the nucleus. In the nucleus, you have the GEF enzyme, and the GEF enzyme will replace the GDP with the GTP, and with the GTP bound Ran is transported into the cytoplasm, and once it is in the cytoplasm, you have the next enzyme, the GTPase enzyme, which will remove the GTP to a GDP, converting that into a GDP, and this cycle continues. So if the Ran molecule decide a protein has to be transported to the nucleus or it has to be transported to outside, this has to be kept in mind as we discuss the next mechanism of when we combine with a protein molecule, what is the exact role played by Ran. To simplify, already the diagram is drawn. Let us see the role played by Ran in importing or exporting a particular protein. So this is the cytoplasm and this is the nucleus. Here we have a cargo. If the cargo has got NLS and NLS is highlighted. Now to the NLS, the important proteins get attached, and in this particular important protein, you have the alpha subunit as well as it's a heterodimer, alpha and the beta subunit. Once it is attached, now it is able to be transported into the nucleus. So it is transported into the nucleus. Once it is in the nucleus, so what happens? Now the cargo must get separated. How to separate the cargo? In order to separate the cargo, now comes if the Ran-GTP comes. Now, if the next step is Ran-GTP will combine with this particular complex. Ran-GTP combines with this particular complex, and now what will happen is if the cargo gets separated, if the alpha gets separated, and if the beta with the Ran-GTP forms a complex. Because Ran-GTP potentially it should be able to repel and remove the cargo because of its charge. So you can understand that how each of these units are getting separated. Cargo is the alpha unit and the beta Ran unit is getting separated. This is how if the cargo is brought inside. And we have a very similar mechanism in order to export a particular protein. What happens in order to export a protein is we have a similar exporting. For example, we have a protein here. Assume this is a protein to be exported, and this protein will have a nuclear export signal. This is the nuclear export signal is present. And this export signal is detected by an export protein, exportins. So this is an exportin. Now, in the role of the Ran comes, we know that there is Ran-ATP, GTP is here. Ran-GTP will combine with this. Once the Ran-GTP combines with this, this is taken out. It is transported to the cytoplasm. Now, in the cytoplasm, we have an enzyme known as Ran-GTPase. Ran-GTPase enzyme is there. So, we have said that this is a GAP enzyme. Okay, GAP activating protein. GTPase activating protein. Now, this GAP enzyme will act on this, and it will convert Ran-GTP to Ran-GDP molecule. You see the previous figure correlate with the previous figure. It's converting Ran-GTP to Ran-GDP, therefore it gets separated. And it will release either cargo plus either export protein separated. And this is how exporting a protein to either cytosolic side from the nucleus occurring with the help of Ran protein. Very interesting mechanism. One of the interesting aspects is how they establish the role of Ran protein. There is a Ran binding domain. Now, this Ran binding domain is able to combine with the Ran-GTP. It can combine with the Ran-GTP, but it cannot combine with the Ran-GDP. It cannot combine. Now, this particular aspect is considered in understanding the role of Ran protein. If you look very closely at the RTB protein, it has got two small domains, and these two domains, we are able to make it fluorescent. So in one place, yellow fluorescent protein is attached. The other place, it is cyan fluorescent protein is attached. Normally, if RPD is not bound to GTP molecule, there is a tendency that they will come closer. It will come closer and it will release energy. And that fluorescence can be detected. We know the technique is called FRET. Using FRET, that is fluorescent resonance energy transfer. So that energy transfer, it can be detected in a fluorescent microscope. So that when will that happen? When there is no GTP, which means only it is bound to GDP molecule that can be detected. And now these two colors, it will give a kind of a green fluorescence when they combine together. Now they have carried out this experiment, and in that process what happened is in the cell, the nucleus becomes green, and if the signal is very weak, because they have come together, the signal will be very weak. Now if GTP is bound, if GTP is present, what will happen in that case is this is not able to come together. So in the RBD, these two YFP and YFP and CFP, they will remain separate. They will remain separate, and there is no energy transfer occurring. When there is no energy transfer occurring, what is observed is it will have a blue fluorescence. And you will see that in the nucleus, if the blue fluorescence is about 20 times, 200 times greater than the green fluorescence that is seen in this particular case when it is not binding to GTP. So when it is binding to GTP, you will see a blue fluorescence. So this was an experiment they carried out in order to prove the role of Ran as well as where it is getting concentrated. Now a question for you. This is the experiment. If this is the experiment, how they would have carried out a control? That is for us to investigate. Okay. Now the question is how this Ran-GDP, which is there in the cytoplasm, how it is brought into the nucleus side. How the Ran-GDP is brought. Ran has its own transport mechanism. What happens to Ran-GDP? Let's begin here. Ran-GDP molecule, Ran-GDP. So, in the Ran-GDP molecule has a transport protein, a kind of a signal that is known as NTF2. It's a protein. Okay, NTF2 can get attached. Maybe it is better that we write it like this. So, to this NTF2 is attached, so it becomes with the NTF2. This is a transporting protein. Once Ran-GDP is bound to NTF2, now this is taken inside. So this is the nucleopore, it is brought inside. So here is the Ran-GDP combined with the NTF2, it is linked. NTF2. Once it is in the nucleus, we have an enzyme. That enzyme is the Ran-GEF enzyme. Ran-GEF enzyme will replace GDP with the GTP. GDP is removed. So you have now Ran-GTP. If the Ran-GTP has no affinity towards NTF2. So in the process NTF2 is released. In the process it is released. And you get Ran-GTP and NTF2. Now NTF2 is a small molecule that is able to pass through a nucleopore, and NTF2 will be transported back into the cytoplasm. So this is how the complete cycle occurs in the case of transportation of proteins. It is also important for us to know these are several mechanisms that have been already established. What are some of the theories that says about transporting the proteins to nucleus? Now, so far there is not so much of clarity on how exactly proteins are transported to the nucleus. All these are experiments that shows that there are several proteins are involved in the transportation of proteins to the nucleus. And all these RANs important, all these are essential. The hypothesis proposed about the transportation of protein is very interesting. So according to that, there are several hypotheses. One of the hypotheses says that if you consider, let us look at, this is a nucleopore. So in the nucleopore, okay, the nucleopore. So in the nucleopore we have looked at the nucleopore structure, and we have seen that it has a lot of FG locations, plenty of FG locations, FG locations, plenty. That is the phenylalanine glycine subunits. I mean, dimer is there, two residues. Now, what is likely to happen is, one hypothesis says that if this is the cargo that is bound to the receptor, and we have seen that the receptor should be able to bind to the cargo, as well as bind to the cargo, as well as it should be able to interact with the FG region. Now, this the important has got two sites which are able to interact with the FG region. Now this interaction will result into interacting, that is pushing into the next one, and in this manner the cargo can be pushed inside the nucleus. That is one hypothesis. And the second hypothesis says that if the FG sequences somehow it come closer, it forms a kind of a tunnel, and that helps the cargo to be transported into the nucleus. Another hypothesis is suggested in order to explain the real transportation of protein to the nucleus. So it requires a lot more of research in order to establish the exact transportation of proteins to the nucleus.